Hello everyone, I am Dr. Deepika Malik. Today's topic of discussion is chromatographic techniques, its principle, types, working, application, advantages and limitations. The Russian botanist Mikhail Svet coined the term chromatography in 1906. The term chromatography is derived from Greek word chroma, meaning color, and graphen, meaning to write. Chromatography is an important biophysical technique that enables the separation, identification, and purification of the components of a mixture for qualitative and quantitative analysis. The first analytical use of chromatography was described by James and Martin in 1952 for the use of gas chromatography for the analysis of fatty acid mixtures. A wide range of chromatographic procedures make use of differences in size, binding affinities, charge, and other properties to separate materials. It is a powerful separation tool that is used in all branches of science and is often the only means of separating components from complex mixtures. Now coming to the definitions. Chromatography is an analytical technique commonly used for separating a mixture of chemical substances into its individual components so that the individual components can be thoroughly analyzed. Chromatograph is the equipment or instrument that enables a sophisticated separation by performing chromatography. A chromatogram is the output of the chromatographic analysis. It can be seen as visual spots, as you can see in the first diagram, or as graphical picture showing peaks shown in the second diagram. Visual spots are seen in mainly planar chromatography like thin layer chromatography and paper chromatography, whereas graphical picture is shown mainly by column chromatography and high performance thin layer chromatography. A chromatographer is a specialist performing chromatography. Now, chromatography can be categorized into two types according to scale of operation. First type is the analytical chromatography, which is used to determine the presence or absence, which is the qualitative analysis, and relative proportions of analytes, which is quantitative analysis in a sample mixture. Mainly planar chromatography, that is paper and thin layer, and sometimes column chromatography like liquid chromatography or gas chromatography are used for analytical purposes. It is done in small scale. Sample number and sample volume required are low. Short columns are sufficient of 2 to 4 mm. Products may not even be collected. It can be categorized based on type of analysis as qualitative and quantitative. Whereas preparative chromatography is used to isolate and purify sufficient quantities of a substance from the sample mixture. Column chromatographic techniques like high performance liquid chromatography, that is HPLC, or liquid chromatography or gas chromatography are mainly used for preparative purpose. It is done in large scale. Both sample number and sample volume are high. Long columns are better, which are of 50 to 200 mm. Products obtained are passed on to a fraction collector. So basically, analytical chromatography is only used for the analysis purpose to determine the presence or absence or its tentative quantity present in the sample mixture. Whereas the purpose of the preparative chromatography is to obtain the purified compound in high quantities. Now let us continue with the analytical chromatography's qualitative analysis. The objective of this approach is to confirm the presence or absence of a specific analyte in a test sample. It gives an idea about complexity of the mixture and the number of compounds present. It is used to check purity and identify any compound. It helps in establishing fingerprints or pattern for analytes extracted or purified from the sample mixtures. These fingerprints can be used to check the identity and the purity of the test samples. Test sample is basically the sample mixture which is to be separated. And by fingerprints here, we mean that a specific analyte produces a specific chromatogram showing a specific pattern. To identify a specific compound in the mixture, a known reference compound, that is standard, and the unknown samples are co-chromatographed under identical conditions. Now how do we analyze in qualitative analysis? In case of visual spots on chromatogram, a pure compound is run simultaneously under same conditions along with the test sample mixture. The presence of spots at the same height and of same color indicates the presence of the same compound in the test sample, as you can see in the given image. Here, for example, a sample mixture is run alongside a berberine standard, and as you can see, the development of a spot at same height and same color would indicate that the compound present in the test sample is similar to the berberine. Whereas in case of graphical picture chromatogram, the formation of one peak and the increase of its height in a chromatogram indicates that the unknown may be identical with the reference compound. Qualitative analysis relies on comparing the retention times, that is the time taken by stationary phase for retaining the compound of the peaks in an unknown sample with those of known standards. We will be discussing these terms in future slides in more detail. If the retention time of a peak in the unknown sample is the same as the standard, then a positive indication can be made. The image shown on the right side shows a chromatogram of the sample mixture showing different peaks and a chromatogram of a reference compound or a standard. The presence of peak of same height and at same retention time indicates that the component present in the sample mixture may be identical to the standard. Now the next is the quantitative analysis. The objective of this approach is to confirm the presence of a specific analyte in a test sample and to quantify its amount. Quantitative measurements are based on measurements of peak height or peak area of the peak from a sample with unknown concentration. The area of each peak in a chromatogram can be shown to be proportional to the amount of analyte producing the peak. Quantification of a given analyte is based on the construction of a calibration curve obtained using a pure, authentic standard sample of the analyte. The value of unknown concentration can be deduced from the curve. The image on the right side shows chromatogram of a standard sample as well as an unknown sample. On plotting a calibration curve of the standard at different concentration, the quantity of the unknown sample can be quantified using it. Although the peak in green and red are present at the same retention time, indicating the samples to be identical, but the difference in the peak height and the peak area indicates the difference in the quantities present in the sample mixture. Next, the chromatographies can again be categorized into two types, but this time according to the methods of holding the stationary phase. Now what is stationary phase? It is the phase over which the mobile phase passes in the technique of chromatography. It remains fixed in place. It is a porous solid like silica or alumina that is coated on a solid support in case of planar chromatography, or packed into a column in case of column chromatography, as you can see in the diagram. Mobile phase is the liquid or gas that flows through the stationary phase. It carries the components of the mixture to be separated. As you can see in the diagram, in case of planar chromatography, a closed chamber is used, whereas in case of column chromatography, a column is used. In case of planar chromatography, a solid support is coated with the stationary phase, whereas in case of column chromatography, the stationary phase is packed within the column. In case of planar chromatography, the mobile phase is present at the base of the chamber, whereas in case of column chromatography, the mobile phase flows from the top of the column into the column and then out of the column. In case of planar chromatography, the flow of mobile phase is from bottom to the top, whereas in case of column chromatography, the flow of mobile phase is from top to bottom. In case of planar chromatography, the sample is loaded at the base of the stationary phase, whereas in case of column chromatography, the sample is loaded onto the top of the stationary phase. Now let us discuss both these types one by one in detail. First is the planar chromatography. It is a two-dimensional technique where stationary phase is adhered to a suitable matrix like paper or is coated on a glass or aluminium plate in case of thin layer chromatography. It is used mainly for detection purpose. It is much faster. It helps to study a number of samples simultaneously. It can be categorized into two types as thin layer chromatography or paper chromatography. Now let us first discuss thin layer chromatography. It is one of the fastest, least expensive, simplest and easiest chromatography technique. It is an analytical technique that can quickly and efficiently separate quantities of less than 10 micrograms of material. In TLC, capillary action allows a liquid or mobile phase to ascend a solid or stationary phase coated on a support plate. TLC has many applications in the organic laboratory. It is used for the rapid analysis of reagent and product purity and to quickly determine the number of compounds in a mixture. Also, by comparing an unknown compound's behavior to the behavior of known standard compounds, mixture compounds can be tentatively identified, as discussed earlier. Now coming to the principle of TLC, the principle of separation in TLC is adsorption, that is the process by which a solid holds molecules of a liquid or solute as a thin film. The separation relies on the relative affinity of compounds towards both the stationary and mobile phase. The compounds in the mobile phase move over the surface of the stationary phase. The movement occurs in such a way that the compounds which have a higher affinity to the stationary phase moves slowly, while the other compounds which have lower affinity to the stationary phase travels fast. The rate of migration of compound up the plate also depends on polarity. The most polar compound moves the least due to a higher affinity to the stationary phase, while the least polar moves the most due to a lower affinity. Therefore, the separation of the mixture is attained. On completion of the separation process, the individual components from the mixture appear as spots at respective levels on the plates. Their character and nature are identified by suitable detection techniques. So to summarize, when a sample mixture is applied onto the stationary phase of a thin layer chromatography, the components present in the sample mixture will get adsorbed to the stationary phase depending upon their relative affinities as well as their polarities. The component with higher affinity and polarity will bind to the stationary phase more strongly as compared to the other components and therefore will move up the stationary phase at a relatively much slower speed than other components. This difference in the adsorption power leads to the separation of the components. Now let us understand the working of TLC. Firstly, thin marks are made at the bottom of the plate with the help of a pencil. Sample solutions, usually a few microliters, is applied to the marked spots, also called as origin. Loading must be slow and a period between each application should exist for drying. It means that the sample should not be applied to the plate at once. The sample should be loaded onto the plate slowly and steadily. After that, mobile phase is poured into the TLC chamber and to maintain equal humidity, a moistened filter paper is placed in the mobile phase. The purpose of placing the filter paper is to saturate the chamber because chromatography in unsaturated chambers result in evaporation of the solvent. After that, the plate is placed in the TLC chamber and closed with a lid. It is kept in such a way that the sample faces the mobile phase. Or we can say that the sample origin faces downwards. Care should be taken that the sample spots are well above the level of the mobile phase. It should not be immersed in the solvent. The bottom, usually 0.5 cm of the plate, is immersed in the mobile phase for development and allowed to travel up the plate by capillary action. Waiting is done till the development of spots. The solvent is not allowed to run over the edge of the plate. Once the spots are developed, the plate is taken out and the solvent front is marked immediately. Solvent front is the level up to which the solvent travels up the plate. Next, the solvent is allowed to evaporate completely by drying the plate. The sample spots are then observed. As you can see in the diagram, a TLC plate coated with silica gel is taken. Spotting of the samples is done at the origin. The first sample taken is a known sample, which is also called as standard. It is basically a pure compound, which is taken to compare with the unknown components of the test sample 1 and 2. After all the three samples are spotted onto the TLC plate, it is placed within the TLC chamber containing the solvent or mobile phase at the bottom and a filter paper to maintain the humidity and saturation to avoid evaporation of the solvent. The chamber is made airtight using a tight lid at the top. After some time, the solvent starts moving up the plate and taking along with it different components of the sample mixtures. After some more time, the analytes or components present in the sample mixture get separated based on their difference in the affinities towards the stationary phase. The components which have a higher affinity towards the stationary phase travels a smaller distance as compared to the components which have a low affinity towards the stationary phase. After the spots are developed, the plate is taken out of the chamber and observed for the spots. For a better observation, spots of different colors are used. The pure standard sample will show just one spot because it do not contain any other components. Whereas the test sample 1, which is pink in color, gets separated into its different component, that is blue and yellow component. The blue component, showing a higher affinity towards the stationary phase, travels to a shorter distance up the plate as compared to the yellow component, which shows a relatively lower affinity towards the stationary phase and moves to a much longer distance. Similarly, the test sample 2, which is green in color, gets separated into its other two components, that is light blue and red component. Since the red component present in the test sample 2 travels the same distance as that of the standard sample taken, it can be concluded that the red sample in the test sample 2 is identical to the standard sample taken. Now, one of the most important parameter of thin layer chromatography is the retardation factor, also designated as RF. The fundamental parameter used to characterize the position of a sample zone in a thin layer chromatography is the retardation factor or RF value. The retardation factor can be calculated using the formula as distance moved by the analyte from origin divided by the distance moved by the mobile phase from origin. As you can see in the diagram that the distance moved by the solvent is designated by A, whereas the distance moved by the analytes is designated by B, C, D and E. When RF value is equal to 0, it means that the sample remains at the origin and it didn't move up the plate. And when the RF value is equal to 1, it means that the sample migrates with the solvent front and does not interact with the stationary phase. Further, the bigger the RF value, the further the spot moved by the analyte. That is, the RF value is directly proportional to the distance moved by the analyte in the TLC plate. Also, the RF value should be the same for a component regardless of how far the solvent moves. That is, the movement of a particular analyte is independent of the distance travelled by the solvent on the TLC plate. When the samples are available as standards, it is common practice to separate the standards and samples in the same system for identification purposes, as we have seen in the previous diagram. Retardation factor depends on many parameters such as nature of adsorbent. Different adsorbents will give different RF value for same solvent. Next is the mobile phase. The purity of solvents and quantity of solvent mixed should be strictly controlled. It should be made freshly for each run if one of the solvent is very volatile or hygroscopic. Example, acetone. Temperature. Although precise control of temperature is not necessary, the tank should be kept away from sources of heat, direct sunlight, etc. As the temperature is increased, volatile solvents evaporate more quickly, solvents run faster, and RF value generally decreases slightly. Next parameter is thickness of layer. Standard plates approximately 250 micrometer is the preferable thickness of layer. Below 200, the RF value vary considerably. Next is developing tank or chamber. It is important that saturated conditions are attained for running TLC plates. This is best accomplished by using small tanks with filter paper liners and sufficient solvent, and by leaving the tank to equilibrate for at least 30 minutes before running the plate. A well-fitting lid is essential. The next parameter is the amount of sample. Increasing the mass of sample on the plate will often increase the RF. Now let us discuss the steps of TLC one by one in detail. First is the preparation of TLC plates coated with the stationary phase. Then is sample preparation and spotting of the sample. Third is the mobile phase. Fourth is the developing the plate. Fifth is drying the plate. Sixth is detection of spots. And seventh is isolation of purified compounds. Coming to the first step of TLC, that is preparation of TLC plates coated with stationary phase. Glass or plastic plates coated with silica gel of particles in the range of 2 to 25 micrometers are used, which are chemically inert and stable. By inert here we mean that a substance should not be chemically reactive. TLC plates are usually commercially available with standard particle size ranges to improve reproducibility. They are prepared by mixing the adsorbents such as silica gel, alumina, kisel gur or cellulose with a small amount of inert binder like calcium sulfate or gypsum and water. Fluorescent additives are added for labeling the adsorbents in case for detection of separated spots by UV light. This mixture is spread as thick slurry on an unreactive carrier sheet, usually glass, thick aluminium foil or plastic. The resultant plate is dried and activated by heating in an oven for 30 minutes at 110 degrees Celsius. The thickness of the adsorbent layer is typically around 0.1 to 0.25 mm for analytical purposes and around 0.5 to 2 mm for preparative TLC. Next step is sample preparation and spotting of the sample. For sample preparation, the solvent used for dissolving sample should be volatile, so that when the sample is applied on the TLC plate, only the sample should remain at the origin and the solvent should evaporate and should not interfere with the process. Next, the sample should be dissolved in a non-polar solvent, as polar solvent has a tendency to spread out at the starting spot as it will get absorbed by the stationary phase. After the sample is prepared, next step is their spotting on the plate. Spots are made using pencil. The thin end of the spotter is placed in the dilute solution. The solution will rise up in the capillary tube by capillary action. Capillary action describes the ability of a liquid to flow against gravity in a narrow space such as a thin tube due to adhesive and cohesive forces interacting between the liquid and the surface of the tube. The plate is then touched briefly by the sample-filled capillary tube at the start line. The solvent is then dissolved to evaporate and again spotting is done at the same place. This way we will get a concentrated and small spot of the sample mixture. Spotting too much material is avoided because this will deteriorate the quality of the separation considerably, which will lead to the tailing of the sample on the TLC plate. Now after the preparation of the plate and spotting of the sample, next part is the mobile phase. Mobile phase is the one that moves and consists of a solvent mixture or a pure solvent. This phase should be particulate free. The higher the quality of purity, better is the development of spots. For silica gel chromatography, the mobile phase is an organic solvent or mixture of organic solvents. The strength of a mobile phase depends on the particular solvent used. The interaction of analytes is based on the like dissolves like principle. The mobile phase is always less polar than the stationary phase in TLC. Therefore, polar compounds will tend to have a lesser affinity for the mobile phase than non-polar compounds. Thus, polar compounds will travel slower up the plate and have a low RF value, and non-polar compounds travel faster up the plate and have a high RF value. What if more polar solvent is used? The more polar the solvent or solvent mixture, the further it will move a polar compound. Polar groups in the solvent can compete with the analyte for binding sites on the surface of the silica gel. Therefore, if a highly polar solvent is used, it will interact strongly with the surface of the silica gel and will leave few sites on the stationary phase free to bind with the analyte. The analyte will therefore move quickly past the stationary phase. Similarly, polar groups in the solvent can interact strongly with polar functionality in the analyte and prevent interaction of the analyte with the surface of the silica gel, which we don't want. With non-polar compounds, there will not be a marked increase in the distance migrated with increasing mobile phase polarity because they migrate towards the solvent front. Thus, we learn that there are certain disadvantages of using a more polar solvent in TLC. Now, next step is the developing the plate. A closed chamber is used to develop plates. It is responsible to keep a steady environment inside, which will help in developing spots. Also, it prevents the solvent evaporation and keeps the entire process dust-free. A small amount of solvent, that is mobile phase, is placed in the container. The solvent level has to be below the starting line of the TLC, otherwise the spots will dissolve away. A filter paper has to be placed inside the chamber. It is moistened in the mobile phase. This is done to saturate the chamber to avoid evaporation of the solvent, especially near the solvent front of the plate. After development, the plate is removed from the chamber and the level of solvent front is marked using a pencil. The plate is then dried in oven to evaporate the solvent. Now after the plate is dried, the next step is detection of spots. Detection can be done by physical or chemical methods. Physical methods does not change the substance on the plate, so it can be used for further study. Physical method includes fluorescence in UV light. Here, silica gel impregnated with fluorescent material is often used to prepare TLC plates in order to observe the UV absorption by an analyte. Developed plate is irradiated with light at 254 nanometer. UV absorbing species appear as a black spot on a yellow-green background. Another method is natural fluorescence, where the samples are irradiated with light at 365 nanometer. In chemical methods, staining of separated analytes is done either by dipping a plate into a developing reagent or by spraying the reagent on it. Then the plate is dried and a distinguished color is developed for an analyte. Many reagents exist that react with various solutes on a TLC plates to yield colored or fluorescent species for visualization. Iodine, most commonly used, produces a light brown spot with most of the compounds, as you can see in the image. Ninhydrin for amino acids is used, rhodamin B is used for lipids, antimony chloride for steroids and terpenoids, sulfuric acid with heating for any organic compound that produces charring. Charring is producing of a blackened spot due to partial burning. Potassium permanganate with sulfuric acid is used for hydrocarbons. Acetaldehyde and sulfuric acid for carbohydrates. Bromine vapors for olefins. Dragendorf reagent for alkaloids. These physical and chemical methods are usually employed for the plates where non-visible spots are present after developing the plate. Basically for colorless spot, whereas colored substances can be seen directly after the plate development. Now the next step includes the isolation of purified compound. While detecting under UV light, the bands are marked with the pencil corresponding to the compound we wish to isolate, and then the silica of said bands is separated by carefully scraping the plate with a spatula, as you can see in the image. The silica of each band is collected separately in a flask and the compound is extracted with methanol by stirring magnetically for 10 to 15 minutes. The solvent is then filtered and evaporated. The residue corresponds to the band or compound scraped out from the silica. In the next slide, we discuss a variant of TLC, that is high performance thin layer chromatography, also designated as HPTLC. It is an extension of TLC, which is robust, simplest, rapid and efficient tool in quantitative analysis of compounds. HPTLC is an analytical technique based on TLC, but with enhancements intended to increase the resolution of the compounds to be separated and to allow quantitative analysis of the compounds. The process steps of HPTLC are identical to classical TLC. The main difference between them is in the characteristics of the separation plate. HPTLC plates are based on optimized silica gel 60 with a significantly smaller particle size than used for classical TLC. This allows a higher packing density and a smoother surface. Hence, the sample diffusion is reduced, resulting in compact bands or spots. Furthermore, the smaller particle size and thinner layer significantly increases detection sensitivity and analysis speed. High throughput analysis using HPTLC is being aimed at the rapid analysis of large numbers of compounds. Only 3 to 20 minutes for optimal separation is required. 5 to 10 times better detection sensitivity than classical TLC. Sample is loaded using an automated syringe on pre-coated plates. The intensity of each positive band increases in proportion to the concentration of the compound present. The first image on the right side is of HPTLC equipment and the second image shows a highly resolved HPTLC plate. The table shows basic differences between TLC and HPTLC. TLC is a manual technique whereas HPTLC is an instrumental based technique. TLC has a low efficiency whereas HPTLC have a high efficiency. The mean particle size of stationary phase in TLC is usually 10 to 12 micrometer, whereas in HPTLC it is much lesser, about 5 to 6 micrometer. TLC plates can be made in laboratory or are available commercially as pre-coated, whereas HPTLC plates are available pre-coated only. Thickness of stationary phase is usually 30 micrometer in TLC plate, whereas about 12 micrometer in HPTLC. Spotting of sample is manual in case of TLC by using a capillary tube, whereas in HPTLC it is automatic by the use of syringe. Now coming to the applications of TLC. The qualitative testing of various medicines such as sedatives, local anesthetics, anticonvulsant tranquilizers, analgesics, antihistamines, steroids, hypnotics is done by TLC. TLC is extremely useful in biochemical analysis such as separation or isolation of biochemical metabolites from its blood plasma, urine, body fluids, serum, etc. Thin layer chromatography can be used to identify natural products like essential oils or volatile oil, fixed oil, glycosides, waxes, alkaloids, etc. It is widely used in separating multi-component pharmaceutical formulations. It is used to purify any sample and direct comparison is done between the sample and the authentic sample. It is used in food industry to separate and identify colors, sweetening agents and preservatives. It is used in the cosmetic industry in the identification of dye raw material and end products, preservatives, surfactants, fatty acids and constituents of perfumes. Now let us discuss the advantages and limitations of TLC. It is a simple process with short development time. It helps in visualization of separated compounds spot easily. The method helps to identify individual compounds. It helps in isolation of most of the compounds. The separation process is faster and the selectivity for compounds is higher. The purity standards of the given sample can be assessed easily. It is cheaper than other chromatographic techniques. It is good for detection of trace impurities. It can detect almost any compound including organic materials. The spot can be viewed by general or specific reagents or UV fluorescence. Pure compounds can be easily retrieved for subsequent structure confirmation. Limitations include that TLC plates do not have longer stationary phase. When compared to other chromatographic techniques, the length of separation is limited. The results generated from TLC are difficult to reproduce. Since TLC operates as an open system, some factors such as humidity and temperature can be consequences to the final outcome of the chromatogram. The detection limit is high and therefore if you want a lower detection limit, you cannot use TLC. It is only a qualitative analysis technique and not quantitative. Only its variant HPTLC can be used for quantitative analysis of compounds. Now the second type of planar chromatography is paper chromatography. Paper chromatography is a type of a planar chromatography whereby chromatography procedures are run on a specialized paper. Paper chromatography is considered to be the simplest and most widely used of the chromatographic techniques. This technique uses paper sheets or strips as stationary phase through which a solution is made to pass. It is an inexpensive method of separating dissolved chemical substances by their different migration rates across the sheets of paper. Now let us understand the principle of paper chromatography. The main principle involved is partition, wherein the substances are distributed or partitioned between liquid phases. One phase is the water, which is held in the pores of the filter paper used, that is cellulose paper, which acts as stationary phase, and the other is the mobile phase, which may be an organic solvent or buffers which moves over the paper. The mobile phase or developing solution travels up the stationary phase carrying the sample with it under the capillary action of pores in the paper. Components of the sample will separate readily according to how strongly they adsorb onto the stationary phase versus how readily they dissolve in the mobile phase. As discussed earlier also in TLC chromatography, the components which get adsorbed strongly onto the stationary phase will travel slower, whereas the components which do not get strongly adsorbed by the stationary phase will move faster. The principle can also be adsorption between solid and liquid phases, wherein the stationary phase is the solid surface of the paper and the liquid phase is of the mobile phase. Such chromatography is termed as paper adsorption chromatography. But most of the applications of paper chromatography work on the principle of partition chromatography. Now let us see the working of paper chromatography. Thin marks are made at the bottom of the paper with the help of a pencil. Sample solutions, usually a few microliters, is applied to the marked spots, also called as origin. Loading must be slow and a period between each application should exist for drying. Mobile phase is then poured into the developing chamber. The inner wall of the tank is wrapped with a filter paper to keep the tank saturated. The paper is placed in the chamber and closed with a lid. It is kept in such a way that the sample faces the mobile phase. Sample spots are well above the level of the mobile phase. It should not be immersed in the solvent. The bottom, usually 0.5 cm of the paper, is immersed in the mobile phase for development and allowed to travel up the plate by capillary action. Waiting is done till the development of spots. The solvent is not allowed to run over the edge of the paper. Once the spots are developed, the paper is taken out and the solvent front is marked immediately. Next, the solvent is allowed to evaporate completely by drying the paper. The sample spots are then observed. The image on the right side illustrates the apparatus for the paper chromatography, where the paper is hung from the top attached to a clip. The main parameter here is also retardation factor, which again is calculated by the distance moved by the analyte from origin divided by the distance moved by the mobile phase from origin. Now let us discuss the steps of paper chromatography one by one in detail. First is the stationary phase and papers. Second is the sample preparation and spotting of the sample. Third is mobile phase. Fourth is developing the paper. Fifth is drying the paper. Sixth is detection of spots. Stationary phase and papers. Fine quality cellulose paper with defined porosity, high resolution, negligible diffusion of sample, and favoring good rate of movement of solvent are preferred. Whatman filter papers of different grades like number 1, number 2, number 3, number 4, number 20, number 40, number 42, etc. are used. In general, the paper contains 98 to 99% of alpha cellulose and 0.3 to 1% of beta cellulose. Other modified papers used for paper chromatography are acid or base washed filter paper, glass fiber type paper, hydrophilic papers, that is papers modified with methanol, formaldehyde, glycol, glycerol, etc. Hydrophobic papers, acetylation of hydroxyl groups leads to hydrophobic nature. Impregnation of or soaked with silica, alumina or ion exchange resins can also be done. Next is the sample preparation and spotting of the sample. If solid sample is used, it is dissolved in a suitable solvent. Preferably, a volatile solvent is used for sample dissolving so that it gets evaporated and do not interfere with the process. After that, sample spotting is done, where sample, usually 2 to 20 microliter, is added on the baseline as a spot using a micropipette and air dried to prevent the diffusion. After that is the mobile phase. Pure solvents, buffer solutions or mixture of solvents can be used. Examples like hydrophilic mobile phase includes isopropanol or ammonia and water in the ratio of 9:1:2, methanol and water in the ratio of 4:1, and butanol and glacial acetic acid and water in the ratio of 4:1:5. Hydrophobic mobile phases includes mixture of dimethyl ether, cyclohexane, kerosene and isopropanol. The commonly employed solvents are the polar solvents, but the choice depends on the nature of substances to be separated. If pure solvents do not give satisfactory separation, a mixture of solvents of suitable polarity can be applied. Different combinations of organic and inorganic solvents may be used depending on the analyte. Example, butanol, acetic acid and water in the ratio of 12:3:5 is suitable solvent for separating amino acids. Also, as you can see in the diagram, that the most polar solvent will move to a lesser extent as compared to the least polar solvent. This is because polar molecules have more affinity towards the stationary phase and therefore travel to a lesser extent, whereas the least polar molecules have less affinity towards the stationary phase and therefore travels longer distances. The next step is developing the paper. The chromatographic chambers are made up of many materials like glass, plastic or stainless steel. Glass tanks are preferred most. They are available in various dimensional size depending upon paper length and development type. The chamber atmosphere should be saturated with solvent vapor. The inner wall of the tank is wrapped with the filter paper before solvent is placed in the tank to achieve better resolution. Chromatography in unsaturated chambers result in evaporation of the solvent from the layer, particularly near the front. Sample-loaded filter paper is dipped carefully into the solvent, not more than height of 1 cm, and waited until the solvent front reaches near the edge of the paper. Now, depending upon the type of paper development, paper chromatography can be of 5 types. First is ascending development. Like conventional type, the solvent flows against gravity. As you can see in the diagram, the spots are kept at the bottom portion of paper and kept in a chamber with mobile phase solvent at the bottom. Second type is descending type. This is carried out in a special chamber where the solvent holder is at the top. The spot is kept at the top and the solvent flows down the paper, as you can see in the diagram. In this method, solvent moves from top to bottom, so it is called descending chromatography. Another type is ascending-descending development. A hybrid of above two techniques is called ascending-descending chromatography. Only length of separation is increased. First ascending takes place, followed by descending. Next type is circular or radial development. Here, spot is kept at the center of the circular paper. The solvent flows through a wick at the center and spreads in all directions uniformly. And the last type is two-dimensional chromatography. Here, the chromatogram development occurs in two directions at right angles. In this mode, the samples are spotted to one corner of rectangular paper and allowed for first development, as you can see in the diagram. Then the paper is again immersed in the mobile phase at right angle to the previous development for the second chromatogram. So basically, the position of same paper is changed twice to achieve the spots on the chromatogram. After the development, the solvent front is marked and the paper is left to dry in a dry cabinet or oven. Next step is the detection of spots. Analytes can be detected by their color. Colorless analytes can be detected by staining with reagents such as iodine vapor, ninhydrin, etc. Radio-labeled and fluorescently labeled analytes are detected by measuring radioactivity and fluorescence respectively. Now coming to the applications of paper chromatography, it can be used to check the control of purity of pharmaceuticals, for detection of adulterants, for detecting the contaminants in foods and drinks, for the detection of drugs and dopes in animals and humans, in analysis of cosmetics and analysis of the reaction mixtures in biochemical labs. Now let us see advantages and limitations of paper chromatography. Advantages include that it is a simple, rapid technique. It requires very less quantitative material. It is cheaper compared to other chromatography methods. Both unknown inorganic as well as organic compounds can be identified by paper chromatography. It does not occupy much space compared to other analytical methods or equipment. It has an excellent resolving power. Limitations include that large quantity of sample cannot be applied on paper. In quantitative analysis, paper chromatography is not effective. Complex mixture cannot be separated by paper chromatography. Less accurate compared to HPTLC, that is high performance thin layer chromatography that we have already seen, or HPLC, high performance liquid chromatography, that we'll be studying in future slides. The next slide shows the comparison between thin layer chromatography and paper chromatography. Thin layer chromatography is based on the principle of adsorption, whereas paper chromatography is based on the principle of partition. The stationary phase for TLC is made up of silica gel or alumina coated on glass plates, whereas in paper chromatography, Whatman filter paper with no coating is used. Amount of sample is more in case of TLC and less in case of paper chromatography. Time taken by TLC is less as compared to paper chromatography. Separation bands are more sharp in case of TLC, whereas in case of paper chromatography, they are less sharp. Sensitivity of detection is high in TLC and less in paper chromatography.